Let's dive in as we're getting closer to lunch. A lot of the methods we developed were to capitalize on being able to look at formalin-fixed paraffin-embedded tissues. They're stored worldwide. There's millions of them out there. And if we're going to look at disease, particularly cancer, changes in cancer glycosylation, these would be the best kind of tissues you could start with if you could make it work. And so, because of course you can immunostain one at a time. You can even do the glycoprotein PSA. This is a prostate. But then we were able to map with the different glycans across the tissue. There'll be a theme all the way through this: location, location, location. We're really effective at mapping different species of N-link glycans. But it's also linked to very specific structures, substructures, including tumor. And it's a term we're calling glycopathology to characterize this. Briefly, we started with the same workflow, essentially, that you would use to do immunohistochemistry on a FFPE tissue. So we didn't want to change anything. We didn't want to change any of the workflows that the pathologist would have to use, too. So we adapted our workflows to what other people already do. So again, just dewaxing and antigen retrieval, instead of adding an antibody, we spray the pangase enzyme directly onto the tissue. It's a very fine molecular coating. It's not liquid in any sense. It looks dry when you apply it, actually. As Montford indicated, but at each location we're generating spectra such that at the end you can get a compilation and essentially do a heat map of every peak location, and that's really what we're looking at with the images. Oh, I skipped ahead. The good part, we're using a 7 Tesla FT in this case. We get very precise masses, and we can assign structures quite nicely. But then, of course, there is the location part. So this was a pancreatic cancer tissue, well annotated, lots of things going on in the tissue. And we started noticing early on that there are different glycans associated with specific subregions. And then you can do the overlays like this, get this kind of map. Also, once they're released, you can do a lot of different validation. There's some CID functions within the FT. Then there's a new instrument coming, which we'll mention briefly, that also be useful. In terms of the human glycome aspect, the method now is, I guess we've been doing this 3 or 4 years with this TM sprayer. It's really introduced a level of reproducibility. And so it really doesn't matter what we put in front of the sprayer, as long as there's tissue, or I'll show you later, it could be serum, a cell. You can get a very reproducible spraying of that, and it's been also quite exportable. We've published the method in the current protocols journal to like a lot of detail if you really want to dig into it. And also, we can then apply these types of other enzymes. So in essence, what we're approaching is we change the signal by changing the enzyme. Using the sprayer then lets us do this quite effectively. And don't have time to go through all of it. My colleagues, Anand Mehta, who's made most of the enzymes we use, and Peggy Angel at MUSC, we've recently formed a company to start working on some of these projects. You'll kind of see at the end where I think this company is going. So for tissues, as Montford said, we've been doing this a while, and numbers are building up, particularly if you're using tissue microarrays, where you can get a lot of patient samples in one slide. And so for prostate and breast, we've done well over 1,000 patient samples. We're getting higher and higher now with liver cancers, with Anand, and many other tissues. I want to show just one example where we made a custom tissue microarray with 20 tumor types. And these weren't for biomarkers, it was just a core where we had a tumor, and then the pathologist selected a region that wasn't tumor, just so we could compare profiling that way. Uh-oh. Where'd it go? Oh, sorry, I've jumped ahead of myself. Just wait for that, it's going to be amazing. Anyway, here's a better data example. We like this coelectrophore colon cancer tissue. You can see the nasty tumor growing into the muscle and growing out from the muscle. And it's got a lot of nice features. Bruker has been quite helpful. We've got a lot of software. This is literally push button now. We can generate the spectra. You find a peak, you press the button, you can generate this image. You can export this in the skills lab analysis software, and now extract out all the intensity data and start doing a lot of stats and comparisons that way. But you can see kind of again, you can generate a lot of different structures. We like this one in particular when we're comparing different tissues because it's segmentation analysis. It'll group what glycans are present within the tissue, and it generally will follow the histopathology quite nicely. And you can dig in within each color and figure out which glycans are present, and almost always there's a biosynthetic theme to it. There'll be like there's a core fucose or not, there's branching, there's, it's fascinating to watch how it actually works. We also have a Rapiflex will get us down into the 10 micron kind of resolution range. The other ones were more in around 40 micron resolution, and you can get these kind of molecular maps. So this is a pretty picture. So what do we do with this information? There's that amazing TMA I was trying to refer to. If we just home in on the higher mass region, and I think this follows up nicely what Mike T-Meyer was talking about in the stem cells. That's what we see in all these 20 different tumor types. We'll see there's a theme, right? There's trisaccharides, tetrasaccharides, branched, but then the decoration seems to vary specifically with tissue type. And then when the tumor type, also that seems to vary too, that each tissue type, when it has a tumor, tends to follow a certain class and grouping. And so we've looked at 20, and almost every single organ's different. It's at every level, whether even just the normal or within the tumor. So at best, we've covered 20% of the targets, if that's probably being liberal. There's a lot of things to look at in the human glycome as far as mapping this out. But we already knew that. In general, we see these kind of trends to summarize that last picture. As it's progressing, in addition to the high mannoses, which I'll come back to, we'll see that, I guess it's almost dogmatic, that increase in branching, but then it really starts changing. Again, every tumor type, it just seems to differ, whether you have it more silation, more fucosylation, bisex, dilacnacs, polylacnacs. It's a diverse area. So let me dive into just one example. Monfort also showed, and of course with the immunoglobulin G profiling studies, glycans can be very clinical, and we're all moving in that kind of direction. At the tissue level, this is what we see routinely in liver cancers. Generally two types. One is illustrated on the left, and that's the type highlighted by that tetraantenary glycan without the fucose, versus the type of tumors we see with the fucose. It's sort of a base comparison. The ones without the fucose are always, they look like this. They're not subtle. They're always tight. They're right on the tumor. It's easy to find them. The 2540 in comparison tends to be much more diffuse. And if we looked at two different TMAs, and this has been published, we looked at two kinds of examples, just general tumor versus cirrhotic versus normal, or tumor normal pairs across all stages. And you can just see visually this tetra without the fucose lights up in only certain ones and then tends not to show up where the 2540 is present. So there's a differential case there. Same when you look at the normal tumor pairs, there's quite a difference. And now we're getting into quickly at the pathology level. These aren't the same. Outcomes aren't the same. Treatment decisions may not be the same. So we're quickly getting into, how can you apply, whether that, in this case, 2393 is present or not, is going to start driving clinical decisions. This may seem like a weird skip, but there's a new instrument that was released less than about 3 weeks ago. This is a timsTOF Flex. It does incorporate ion mobility, but it's essentially a MALDI QTOF. So everything I've shown you what we can do with the FT and the RappiFlex, it's essentially the RappiFlex laser. It's very high resolution. We can do a 20 micron image in about an hour, hour and a half. It's going to open up the door for 3D, many other applications, and a lot more throughput, and get the benefit of the resolution. And it has ion mobility separation, so we know how important that's going to be for isomers and glycans. And it has a QTOF on the back end, so we can do some sequencing. And I also think it's going to be great for glycopeptides, at least the abundant ones initially. So that's coming. I do have, this is one of our favorite tissues, because it's not subtle. This is a liver cancer. There's either tumor or not tumor. And when we ran it on the timsTOF Flex, we get very distinct regions. We got about the same mass profile that we can get with the FT. In general, on this one, we get about 70 glycans, but you can get some really nice, that's that tumor normal interface, and lots of nice overlays. This was that glycan, or this prostate tissue that I showed up front with the high mannose. And what we do see with the high mannose is that we looked across many, many different tumor types. In prostate, yes, we see lots of high mannose in the tumor, but we also see lots of high mannose in hyperplasia regions. If we look in colon, we'll see them in adenomas. There's other tumors where it's only showing up in the tumor. So it's kind of case by case how these high mannoses. In no scenario do they seem to be good indicators of something good happening. Hyperplasia, tumor, so it's something we're keeping an eye on. We've also got some initial ion mobility data. You can separate the high man isomers, and that's a whole other thing that might be interesting. Bruker likes this image now because it's for people with different degrees of color blindness. They like this. I'm still trying to get used to it, but we'll see. So it's hard to say, be presumptuous to say that we're looking at any masterpieces yet on our images, or are we? Segue. So let me end with a few examples. Once we had nailed down the method, and this was very reproducible as far as the signal, and with the Pengase we're using, it's essentially if we can get it on to the whatever we're trying to digest, we're going to see signal. And it isn't like you get something that's unexpected. That's the beauty of the N-links. There are many ways, there are many variations, but there's only so many ways you're going to see in the mass spec, particularly in the lower mass ranges. So we've just recently adapted this to an antibody slide array. So this is old technology, antibodies to serum glycoproteins. If you put them on the array and then pull them down with, just add serum. So we know where the antibody was on the slide because that's where we put it. So now we know where the target, what was captured also. So then we spray it solid phase like it's tissue. And so therefore there's no migration away from the spot. And so it's essentially an imaging mass spec workflow. We can also use the software in the back end to then quantitate the results. And so this was just a quick example. We beat this to death, I should say, my student Allison Black did an extensive amount of work to prove the specificity of this, which I'm only summarizing in one slide. But it was just published, so you can check out to see how we navigated this to get that. But realistically, if we can do 2, we can do 4. If we can do 4, we can do 8. I think we're up to 8 right now as far as that's the easy button. The kind of slide Monford showed with let's say the top 20 or 30 serum glycoproteins, we're going to easily get this on and profile, essentially glycoprofile each one of those samples and start doing that sort of amazing calculation of how each individual glycoprotein is changing with a disease state. There's a throughput component there that we're still working on also, but it doesn't take that long. Really, the biggest time is the incubation on the slide. We're going after a lot of targets this way because Anand has patented a long time ago that fucosylated glycoproteins in liver cancer. So we're looking at a lot of different commercial possibilities with this. And if we can do an antibody capture, what if you just spot serum? This isn't that trivial because you have to keep it there. So we've been messing with how you get it onto the slide. But once we keep it on the slide, then we just pretend it's tissue. And we can get a good distribution. 6 hours, that's being conservative. We think we can get this into a much even faster clinical range with some temperature incubations in probably 2 hours from application to data acquisition. And so this was a liver cancer example. And this is part of where we're going with our studies, is to try to figure out, can we look at the serum glycans? Those are the same two I was talking about in the liver cancer tissues. Can we start putting them back on the carriers and use that as a diagnostic? And then this is old history for me, but looking at prostatic fluids, again, we just spot it, we can spray it, we can start getting glycan profiles. That's another area I'll go back to. And I'll just end briefly. Peggy Angel has developed an on-slide method, essentially just growing the cells on a slide. And then you fix them, and then we can spray them. And so she does have single-cell glycomics, because you can just literally hit it with a laser and that single cell and get a profile. So I don't know if how that counts, but and it was an endothelial cell, so this wasn't a challenge. So they were pretty big targets. But she's submitted this. There's a lot going on here that she's also adapted into SILAC, but based on time. I just want to highlight that essentially we're working in that direction. And really this is where I'll end. This is our vision component. I think it integrates well with the Human Glycome project. That for an individual sample, and again, particularly on a cancer patient aspect where we're going to have tissue at some point, linking the N-glycan imaging as well as other targets, but then doing blood. We could do total glycan, antibody arrays, immunoglobulin arrays, all of the arrays, but also immune cells themselves probably stuck to a slide. And what if you do essentially ending up with a patient-specific clinical glycotyper test that, we've been hearing it more and more, the whole meeting, these are important and they're telling you stuff clinically. So we think we got a workflow to really get this into the clinician's hands at some point. And just a quick acknowledgement of the group and these people. And again, appreciate your time and the opportunity to present. Thanks.